Hello everyone. Today we're going to be going over muscular tissue. So the purpose of this chapter is going to be learning about the structures in muscle. So the cells, the fibers, and how they work with one another. We're going to look at the events in contraction when it comes to the neuromuscular junction. We're going to describe the reaction by which muscle fibers produce ATP, or adenosine triphosphate, understand the structure and function of motor units and how muscle tension is controlled, and then we're going to compare structure and function of three types of skeletal muscle fibers. So this part, we're going to be really talking about the types of muscular tissue, and then the components of muscular tissue, and then start to talk about contraction. So the three different types of muscular tissue that we have is skeletal, cardiac, and smooth. So you can look at this anatomy overview link, but here's a really good indicator. For skeletal muscle, as we've been kind of looking at the tissues, with the name, they're going to be attached to the skeleton, they're going to be attached to the bones, because their main function is to move those bones, is to give us movement, to give us ability to change our bodies, or not like internal arrangement, but how we interact with the world, basically. And then the appearance of this, it's going to be multinucleated. So you're going to see multiple nuclei, and it's going to be striated. And that's because of the how the fibers and proteins are laid down. It gives that type of appearance. The control is going to be voluntary. This is the one that we think about to move. We don't think about pumping our heart. We don't think about our GI tract, like our stomach or small intestine, large intestines, to work. We don't think about the smooth muscle within our arteries or veins to dilate or constrict. Those are involuntary. And so another thing with the next type of tissue is cardiac. So this is heart tissue. Its main function is to pump blood, and you're going to see one nucleus, usually per kind of area. It's going to be much more, it's going to still have striation, but it's going to have much more of these intercalated discs that you don't see anywhere else, and that's because they are very important for conduction and propagating the signal that it gets. The last one is going to be visceral or smooth muscle. This is going to be in various organs. The example that I've already mentioned is the GI tract. Various functions, so peristalsis is the movement and slow kind of contraction and relaxation that our GI tract does to push things along. Our stomach churns, so it kind of mixes things up to get the acid everywhere, and it does that by kind of contracting in different areas to keep it kind of pushing. The appearance is going to be one nucleus. You're not going to see any striations, and again, this one is voluntary as well. So the functions of muscular tissue, it's going to be producing body movements, stabilizing body positions, storing and mobilizing substances within the body, and then generating heat. And so if you could actually, when we talk about functions, this is kind of the big one. Storing and mobilizing substances within the body is what cardiac and smooth muscle does. Almost everything else is mainly skeletal muscle, but the storing, mobilizing substances within the body is going to be cardiac, and smooth. Properties of the muscular tissue. So they can be electrically excited. So this is why if you get one of those kind of electric pads and you can stick it on your muscles, and it would contract it for you, and you can kind of feel the muscle twitching. That's that electrical excitability. That leads to the contractibility or contraction, or bringing both ends of the muscle together so that it can produce movement. It's also extensible, so that means it can get stretched out. And then elasticity, that it kind of comes back to its normal phase when you stretch it out. So let's continue on with the level of organization. So this is going to be a good kind of way to come back in terms of summary. So when we have the whole muscle, this is going to be one organ. So if we're thinking about our bicep muscle in our arm, right, that's that muscle that is needed for us to do bicep curls. This is that individual muscle is the organ. The muscle fiber is the cell of the organ. A myofibril, myo meaning muscle, fibril meaning fiber. It's a specialized intracellular structure. And then we have myofilaments, which are the filaments within these myofibrils. And then we have the specific protein molecules, myosin and actin, that are going to be acting on these filaments and interacting with it. So here a couple, when we're looking at the bigger ones, these are just a little bit more specific. So we can see here's the bone, here's the tendon that's attaching muscle to the bone, and here's that skeletal muscle that is the organ. We're going to then divide that up, what we call a muscle fascicle. And this muscle fascicle is just a big bundle of muscle fibers. And what we can look at are these muscle fibers right here. And we'll talk about endomysium, paramysium in just a little bit. But as you can see, they're organized in a way that they're compartmentalized so that they can stay in these very certain or strict kind of fiber lines, so that they're doing the best job that they can in the orientation that they're laid down. Next, we have the muscle fiber. This is where we can see all of these smaller myofibrils that are within the muscle fiber. And then when we look at the myofibril, this is the individual unit that contains multiple sarcomeres in a line that are going to be the fibers and filaments and proteins that are needed to actually contract and overlap on one another. And then the filament, or the myofilament, is the contractile proteins. So here's just the bigger view of it. Again, the big things that you kind of need to know here. Let me see if I have. No. Okay, so I had a summary slide. I apologize. So now that kind of goes through the level of organization for this. Now, the things that you need to know are the epimysium. So this is the anatomy portion. Epimysium is going to be surrounding the muscle itself. This is going to be kind of the casing If we're thinking about almost like a sausage or something, there's that really big casing around it, keeping it really compact so that these muscle fibers aren't kind of getting allowing to go in different directions, because then that's when we lose some of its efficiency. When we go down further, we're going to see within this muscle fascicle that there is going to be endomysium that's going to be surrounding a muscle fiber. And then we see perimysium that's going to be around that muscle fascicle. So let's look at this again. So epimysium is around the entire muscle, the very outer area. When we go in and look at a muscle fascicle, this is it zoomed up where we see the muscle fiber. So in the muscle fascicle, we have perimysium, and then around the muscle fiber, we have endomysium. So muscle fascicle, perimysium, muscle fiber, endomysium, and the muscle itself, epimysium. Next, when we look into the muscle fiber, we will see some very specific structures that we'll actually talk about in the next slides. Again, this is just so that you can get the organization aligned in your head, and going from the larger structure down to the smaller structures. So when we look at the microscopic view of a muscle fiber, we really the important things that we need to kind of see are going to be the structures that are connecting it to the other muscle fibers, and that's important because we don't want just one muscle fiber to be acting. We kind of want all of them to be acting at once when we need it, so that we can get that signal out and actually use that muscle right away when we want to. And so those things are called the transverse tubules, or the T tubules, that you can see on this blue. That's going to be connecting with these structures, which are the sarcoplasmic reticulum. And that sarcoplasmic reticulum is going to be important when it comes to releasing calcium. So you've probably remember the endoplasmic reticulum. This is a special kind of reticulum. Instead of making proteins, it's actually going to be storing calcium, and calcium is going to be a big signaler. When it gets released, it will interact with certain things and change the actual structural conformation of proteins. So the sarcolemma, it's a plasma membrane of the muscle cell. So that's going to be this blue casing around the muscle fiber itself. Or, sorry, the myofibril. Or, I was correct. Muscle fiber itself that contain the myofibril. The T tubules are the invaginations of the sarcolemma, so they're just they have the same coating, or it's the same. It's made up of the same thing as the sarcolemma. It just goes into the cells so that it can get into more connections with that sarcoplasmic reticulum, so that we release calcium. Sarcoplasm is the cytoplasm of the muscle fiber, and then myoglobin. So if you know about or remember me talking about hemoglobin, hemoglobin is going to be within the red blood cell. It's a protein that is the functional unit that binds to oxygen and allows our red blood cells to actually move oxygen. And so instead of hemoglobin, it is myoglobin. It's a red colored protein that binds oxygen, found only in muscle. So maybe when you think about when you order a steak and it kind of comes out more like red and pink, and you're like, oh gosh, or when it comes in the packaging, you're thinking, oh, there's all this fluid. They didn't get all the blood out. Well, actually, they did get all the blood out. They're required to get all the blood out. What that is the myoglobin. So it looks red because of the globin, the part that is very similar to the red blood cells that binds oxygen. So here are the big things I wanted to kind of show you, and let me highlight some more. So we have the sarcolemma that you need to know. That's going to be the casing. We have the individual myofibril, which is the unit. Sarcoplasm is going to be just the cytoplasm. That's going to be in the space all around. Here, this transverse tubule, or the T-tubule, is the blue part right here. And you see it invaginating from that sarcolemma. And then you'll see a lot of mitochondria because we need a lot of ATP for the function of these cells. And this is just another kind of view of the cell itself. We have these T-tubules that are invaginating down so that it gives us more surface area and more connection with these sarcoplasmic reticulums here. And then in the main body of it, we have these myofibrils just running along in the cells. And we have multiple nuclei that are making and giving making RNA to send out to make more protein. So when we go down into the myofibril, so right here, here's our myofibril. We're going to zoom into it, and then we're going to look at one of these specific filaments that we see here, and one unit that we see and we call sarcomere. This is the functional unit of the muscle that is going to be causing the contraction of the muscle itself. So when we look at this, we have multiple players that are important. This sarcomere is the one that's that we call the specific unit between the two Z discs, and we're going to have these areas of overlap of thick filaments, this darker red, and this black line, and that is the thin filaments. The interaction of those two is what causes them to come closer and closer, or to go further apart, and that's what allows for the contraction relaxation that we see on the larger level when we actually move our hand or our arm or any muscle. So what we have here are the specific units. We have the thin filament because it looks thin. Then we have the thick filament because it's thick. And because of the protein, the specific ones, we have actin and myosin that are going to be the ones interacting with one another. So actin is going to be on the thin filament. Myosin is going to be on the thick filament. So when we actually got some kind of views of it before we had kind of all these cartoons and illustrations, all we could do is kind of take a slice of it, put it under a microscope and look at it. And this is what we saw. We saw and we labeled these specific visuals here. So we have these things called the z-disks, and they actually do run in kind of a zigzag. These are the narrow plate shaped regions of dense material that separate one sarcomere to the next, and they're kind of an anchor point. They're an anchor point for our thin filaments out here. And then when we look at the middle line, we're going to see an M line, and it's easily called the middle line. Easy way to remember the M line. And at the ends, we have the Z's. So middle is M, at the end, it's Z, just like the alphabet. So now we go into these things called the bands, and we go about what they look like, and then we can associate with what filaments and structures do we see in these bands. I like to start with the I band. The I band is going to be the one that we contain only thin filaments on the ends. So we have two I bands here that we can see. The end of each sarcomere. And then we're going to have the A band. The A band is the overlap that contains thin and thick filaments. Okay, so it contains this overlap right here that has both the thin and thick filaments, and it contains the H band. And I think of the H band as the heavy band because that's where all of the thick and heavy filaments are. So let's go back to this visual. Just kind of look at it. We have our thick filaments here, and we have our light filaments here. Here's our overlap. Okay, and it connects with these brackets here. This overlap and all of the thick filament is the A band. The part that we can only see that we can see with only the thick filaments is the H band. So these are kind of important because it allows us to know where structures are, what's moving, and where things are going. Because what happens, and we'll get into contraction in the later parts of this chapter, is these thick filaments are going to be pulling, kind of like pulling on rope on these thin filaments, and these thin filaments move towards the middle line, the M line, that then we see that bigger kind of contraction on the bigger level. And the proteins that we need to know are going to be the contractile ones and the regulatory ones. Contractile ones are the ones that are important, that are actually interacting and causing chemical reactions to have a movement. Okay, that's why it's contractile. So we have the myosin heads of the thick filament, and we have the actin parts of the thin filament. Okay, actin is going to be kind of bound up and wound within two other structures. it's going to be wound up in this tropomyosin. And the tropomyosin is going to be kind of covering the part of the actin that will actually interact with myosin, because we only want to actually contract when we want it to. If this is always off, we're going to be contracting way more than what we want. So, and we wouldn't be able to relax that well and stretch. And so what we want is this tropomyosin that's going to be covering it. And then we have this protein called troponin. This troponin moves the tropomyosin. Okay, so tropomyosin is covering actin. Troponin is going to be moving the tropomyosin. And you don't need to know these structural proteins. So these are just going to be some summary statements for these proteins, and they have more of the specifics. I'm not going to talk about the specifics because it will be added on to the later portions of this chapter, but so that it's always concise, and when you look at these information that it kind of contains what you need. You don't need to know this. Those are just the structural ones. So in the next video, we'll talk about the contraction and relaxation. This is going to be that's going to be the physiology portion of the chapter.